The clinical presentation of a patient infected with Staphylococcus aureus often begins with a breach in the cutaneous barrier, leading to a localized inflammatory response characterized by the formation of an abscess or furuncle. When this gram-positive coccus is isolated from a clinical specimen and streaked onto a blood agar plate, the resulting morphology provides critical diagnostic clues for the United States medical licensing examination. Staphylococcus aureus is a facultative anaerobe that appears as large, smooth, creamy, gold-colored colonies. This characteristic pigmentation is attributed to staphyloxanthin, a carotenoid pigment that acts as a virulence factor by inactivating the microbicidal effects of reactive oxygen species like superoxide and hypochlorite within host neutrophils. Upon closer inspection of the medium, one observes a distinct zone of clear beta hemolysis surrounding each colony. This complete lysis of erythrocytes is primarily mediated by alpha-hemolysin, a pore-forming toxin that inserts into host cell membranes causing an influx of ions and subsequent osmotic rupture. This distinguishes it from the partial green-hued alpha-hemolysis seen in Streptococcus pneumoniae, or the gamma-hemolysis of Enterococcus. Biochemically, this organism is catalase positive, which allows it to convert hydrogen peroxide into water and oxygen, thereby surviving the oxidative burst of phagocytic cells. This property helps differentiate all Staphylococcus from the catalase-negative Streptococcus. Furthermore, Staphylococcus aureus is coagulase positive, meaning it produces an enzyme that converts fibrinogen to fibrin. This fibrin coat serves as a protective shield against phagocytosis and helps sequester the bacteria within an abscess. Protein A is another vital virulence factor displayed on the bacterial surface, which binds the constant fragment portion of immunoglobulin G at the 17th position. This effectively flips the antibody upside down, preventing opsonization and complement activation via the classical pathway. In the context of toxicology, the organism may produce toxic shock syndrome, toxin 1, a superantigen that cross-links the variable beta region of the major histocompatibility complex class II on antigen-presenting cells with the alpha chain of the T-cell receptor. This bypasses normal antigen processing and leads to a massive release of cytokines, including interleukin-1, interleukin-2, tumor necrosis factor alpha, and interferon gamma. Clinically, this manifests as high fever, a diffuse erythematous rash resembling a sunburn that later desquamates on the palms and soles, and multi-organ failure. For the boards, it is essential to remember that this bacterium is a leading cause of acute endocarditis in intravenous drug users, typically involving the tricuspid valve, as well as the most common cause of osteomyelitis in the general population. Sensitivity to nafcillin is determined by the binding affinity of penicillin-binding proteins, but resistance in methicillin-resistant strains is mediated by the acquisition of the MECA gene, which encodes a modified penicillin-binding protein 2A with low affinity for beta-lactam antibiotics. Treatment for such resistant strains requires vancomycin, which inhibits cell wall synthesis by binding the D-alanine terminus of peptidoglycan precursors.